Hello, welcome to my YouTube channel. This is Soumya. In this video, we will discuss the one more interesting concept from cell signaling, that is quorum sensing. This quorum sensing is a type of cell signaling, and it is a more specifically, it is a type of paracrine signaling. What is this paracrine signaling? Paracrine signaling, it is nothing but a type of a cell signaling where the cells are communicating which are nearby. It sends an information or a message to the nearby cells. Okay, now what is this quorum sensing? This quorum sensing is the regulation of gene expression in response to fluctuations in cell population density. This point is very important here. Cell population density. So the quorum sensing is going to regulate the gene expression in response to what is that? Cell population density. The quorum sensing is a type of stimulus and a response correlated to population density. So this stimulus and response is depend upon cell population density. And this quorum sensing we can see in bacteria. Okay, quorum sensing is used by most of the bacteria. Okay, the example like gram positive bacteria and gram negative bacteria. This quorum sensing bacteria produces and releases a chemical signal molecules called as autoinducers. What are these autoinducers? As in the name indicates, these are going to release some kind of enzymes, and these enzymes itself helps the autoinducers. Okay, so if you think this is an autoinducers, these autoinducers are going to synthesize the enzymes, and these enzymes will help in the increasing the population of the autoinducers. Okay, these in turn are going to synthesize the autoinducers. Okay, that's why the name auto means self. Okay, so it is going to induce the self number. It is going to produce some enzymes, and these enzymes will help in the increasing the number of the autoinducers. Okay, and for example, as quorum sensing can be seen in bacteria like gram positive bacteria and gram negative bacteria. In case of gram positive bacteria, the autoinducer is oligopeptide. Okay, these are all exam point you have to remember. So the gram positive bacteria have a autoinducer. Okay, that autoinducer is here oligopeptide. And in case of gram negative bacteria, the autoinducer is N-acetylated or N-acetyl homoserine lactone. In short, it is called as AHL. Okay, in short it is called as AHL, and this AHL is a lipophilic. It is lipophilic. As this AHL is a lipophilic, this AHL can lipophilic means philic means loving. Okay, lipo means lipid. So as it is a lipophilic, this AHL can easily cross the plasma membrane. Okay, but this oligopeptide autoinducer, it is not a lipophilic. Okay, so because of that, this oligopeptide in gram positive bacteria needs a transporter. Okay, transporter, like it may be a channel or carrier. What is the use of this quorum sensing? The quorum sensing, as I said, it is a type of communication. This quorum sensing communication in bacteria regulates a diverse array of physiological activities. Means it will help in getting so many functions like symbiosis, competency, antibiotic production, and virulence. Next, and then conjugation, which helps in the motility and sporulation, and also formation of biofilm formation, and then bioluminescence. So to regulate all these physiological activity, the main helping communication is the quorum sensing. So this bacteria can able to sense the surrounding environment and adjust to that surrounding environment through a communication called as quorum sensing. And now I am going to explain the quorum sensing in gram negative bacteria. Quorum sensing in gram negative bacteria. This quorum sensing was first discovered to control the bioluminescence in Vibrio fishery. This fishery is a gram negative bacteria. ಆ್ಯಂಡ್ ಹಿಯರ್ ವಾಟ್ ಈಸ್ ದಿಸ್ ಬಯೋಲ್ಯುಮಿನೆಸೆನ್ಸ್ ಅಂದರೆ ಅದು ಕೆಲವೊಂದು ಅನಿಮಲ್ಸ್ಗಳು ಲೈಟ್ನ ಎಮಿಟಿಂಗ್ ಮಾಡ್ತಾರಲ್ವ, ಗ್ಲಿಟರಿಯಾಗಿ ಕತ್ಲಲ್ಲಿ ಲೈಟಿಂಗ್ ಎಲ್ಲ ಕಾಣಿಸುತ್ತಲ್ವ, ಅದನ್ನು ನಾವು ಬಯೋಲ್ಯುಮಿನೆಸೆನ್ಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಓಕೆ, ದಟ್ ವಾಸ್ ಶೋನ್ ಬೈ ಅ ಗ್ರಾಮ್ ನೆಗೆಟಿವ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಾಲ್ಡ್ ಆಸ್ ವಿಬ್ರಿಯೋ ಫಿಶರಿ. Okay, it was first discovered in Vibrio fishery. So if you take the gram negative bacteria, if you think this is the gram negative bacteria, okay, this is the gram negative bacteria, and it has a genes, okay, it has a genes called as lux R and lux I, okay, and a structural genes called lux C and then lux D and then lux B and then lux A and lux E and lux G. As there is no space, I just wrote it outside. Okay, so these are all C, D, B, A, E, G. It is a structural genes, and lux R and lux I is the regulatory genes. And remember, in one of the exam even they are asked this sequence as well. So what I'm explaining is important. In previous year question paper they asked the gram negative bacteria autoinducer, that is N-acetyl homoserine lactone, that is AHL. Okay, so concentrate here. And here the lux I is going to synthesize a enzyme that is called as AHL synthase. It is going to synthesize an enzyme called as AHL synthase. This autoinducer is going to synthesize an enzyme that is AHL synthase. And this AHL synthase enzyme, as I explained just now, it is an enzyme. Synthase is an enzyme. This AHL synthase enzyme is going to synthesize a autoinducer itself. Means it is going to increase the AHL number. So it is going to increase the AHL number. So inside the bacterium, so there is an increase in the number of AHL. Okay, and as I said just now, this AHL is a lipophilic, and then it is easily can pass through the plasma membrane. It can able to pass through the plasma membrane. So by a simple diffusion. Here what is this diffusion? Diffusion means the movement of molecules from higher concentration to the lower concentration. As this number is increased inside the cell, the AHL is moving outside, okay, to the lower concentration. Okay, so and here, so this is a one bacteria, right? So in outside there will be many bacteria will be there, and these bacteria will also show the same process here. So they will also release the AHL to the surrounding environment, right? So they are releasing the AHL to the surrounding environment. So then here now it becomes there is an increase in the AHL number outside the bacterial cell. Okay, as there is an increase in the AHL number here, so these will in turn because now the concentration is higher here, and here it will become lower, right? So these will again enter into the AHL is going to enter into the or inside the bacterial cell, and this AHL is going to bind to the lux, or it is going to bind to the lux or, okay, regulatory gene. It is going to bind to the lux or a transcription factor. This one is going to it is a transcription factor. As it is a transcription factor, and it is going to activate the structural genes, and it will lead to a synthesis of mRNA. Okay, the transcription process is going to take place, and later this mRNA produces a enzyme called as luciferase. Okay, this luciferase is going to regulate the bioluminescence. Okay, that is in case of gram negative bacteria. Now we will discuss the very important one which has been asked in the examination, that is we will learn the what happens in case of gram positive bacteria. Okay, so in gram positive bacteria, as I just mentioned now, the autoinducer in case of gram positive bacteria is oligopeptide, and this oligopeptide is unable to cross the plasma membrane. Okay, it needs a transporter. Okay, it needs a transporter, or you can say exporter. Okay, you have to remember that point. And this gram positive bacteria, it communicate with a signaling, a type of signaling called as two component signaling. This is exam point and it is very important. So gram positive bacteria communicate with a signaling called as two component signaling. Why it is called as two component signaling means there are two important proteins here. So those two important proteins are sensor kinase. Okay, as in the name indicates sensor, it is going to sense. Okay, and there is a response regulator. Response means it is gives the response to the stimulus. Okay, it's going to regulate the response. Okay, these two proteins are going to help in the communication. That's why these two are called as two component signaling. Okay, important, you have to remember this. Now in case of gram positive bacteria, think this is a gram positive bacteria. Okay, this gram positive bacteria have genes. This gram positive bacteria, the auto-inducer is oligopeptides, right? These oligopeptides are going to produce as the auto-inducers. Okay, these are going to in turn produce as the auto-inducers, and there is a increase in the number of the oligopeptides, right? So, and these are about to cross this plasma membrane, but they are unable to cross the membrane. They require some exporters. Exporters are transporters. Okay, and then later here they are going to transport the outside the plasma membrane. Okay, so when they are entered the outside the plasma membrane, and there is a, I said this is a two component signaling, right? In two component signaling, so on the plasma membrane there is a sensor kinase. Okay, this sensor kinase protein, for this sensor kinase protein, this oligopeptide autoinducer comes and bind to the sensor kinase. Okay, to this sensor kinase, this autoinducer comes and binds here. Okay, and this one is going to phosphorylate the histidine kinase. It is going to phosphorylate the histidine kinase. Okay, it means it is going to add the phosphate group, then it will become activated. So after the activating, there is another one called as response regulator, right? This one response regulator. This response regulator is attached to the aspartate amino acid called as aspartate, which is number that is represented as D. Aspartate, I guess I have given amino acids, 20 amino acids with 3-letter code and single code. Okay, aspartate, it is represented in D. Okay, so this response regulator have aspartate here, and this one is a response regulator. It is a transcription factor. Okay, transcription factor. This response regulator transcription factor, it is going to activate the Lux promoter. It is going to activate the Lux promoter, and this Lux promoter drives the gene expression. This histidine kinase is phosphorylated. Okay, that is it, the transfer of phosphate, that is called as phosphorylation. Okay, so these are all exam point. You have to remember this, how the gene expression is going to take place in the gram positive bacteria. It's my pleasure to share my knowledge with you all. If you like this video, please do share and subscribe. Thanks for watching.